So according to the size of the bacteria, the Bacillus anthracis, which is considered to be the large size, has the size of the Bacillus anthracis is 1 to 1.3 micrometer into 3 to 10 micrometer, and which is compared to the small cells such as Pasteurella tularensis is 0.2 into 0.2 to 0.7 micrometer. Whereas Mycoplasma, which are also known as atypical pneumonia group, that Mycoplasmas are also even smaller compared to the Pasteurella tularensis, and the measurement of Mycoplasmas are 0.1 to 0.2 micrometer in diameter. So, these bacterial cells are measured in the micron or micrometer. Bacteria therefore have a surface to volume ratio that is very high, about 100,000. And these bacteria are stained by the staining reagents and then visualized under high power of magnification, that is 1000x of compound microscope. So using this microscope, we can visualize the bacteria which are stained. An electron microscope was now established, which is used for the clear visualization of internal structure of bacteria. And according to this, after the discovery of this electron microscope, we can correctly measure the size of the bacteria in terms of micron. That is, for example, the size of cocci ranges from 0.3 to 3 micrometer. Then the size of the rod-shaped bacteria, it also ranges from 0.15 to 2 micrometer width to 0.5 to 20 micrometer in length. So these bacteria are measured using a calibrated slide under a compound microscope or by using an electron microscope. While we are focusing the specimen under the compound or electron microscope, we can measure those bacteria using the calibrated slide, and we can measure in terms of micron. And this is the picture of the size of the bacterial cell in the light microscopic view as well as in the electron microscopic view. And there are different types of structures whose size is mentioned, such as atom, small molecules, lipids, proteins, virus, then bacteria, then organelles, then coming eukaryotic cells. So eukaryotic cells are comparatively large to that of the other structures. Next is about the shape of the bacteria. The shape of the bacteria, that is, actually this bacteria and archaebacteria are classified by the direct examination with the light microscope according to their morphology and arrangement. So based on this morphology and arrangement, this bacteria and archaea can be classified. What are the different basic morphologies? Mainly they are spheres, which are also known as coccus. Then round-ended cylinders, which are also known as bacillus. But there may be others such as helically twisted cylinders, for example spirochetes. Then cylinders curved in one plane, that is selenomonads, that is cylinders curved in one plane. And also there are certain structures with unusual morphologies such as square, flat box shaped of archaean genus Haloquadratum. These are the microbes with unusual morphologies. Then first coming is about coccus. So a coccus, the plural is cocci. Singular, it is called a coccus. It has the name coccus, which has been derived from the Latin word coccinus or scarlet and derived from the Greek word coccos, which means berry, because it has a structure resembling berry. Then it is any microorganisms, usually bacteria, whose overall shape is spherical. So it is almost all the coccus are spherical or nearly spherical, describing a bacterium as either coccus or spheres. So this is the first of many taxonomic traits for identifying and classifying a bacterium according to the important human disease caused by the coccoid bacteria. So this is the first of many taxonomic traits for identifying and classifying a bacterium according to the human disease. So the human diseases caused by this coccus or coccoid bacteria are staphylococcal infections, such as mainly the coccoid bacteria are staphylococcus. So the infections caused by this organism staphylococcus are called staphylococcal infections. For example, food poisoning, urinary tract infections, toxic shock syndrome, gonorrhea, as well as some forms of meningitis, throat infections, pneumonia, and sinusitis. So these are the coccus. Then coming is arrangement of cocci. Now, how the cocci is arranged? The cocci bacteria can exist singly, either in pairs, which are called diplococci, or in groups of 4, such as tetrads, or in chains, such as streptococci, then in clusters, for example, staphylococci in the form of grapes, grape clusters. In cubes consisting of 8 cells are called sarcinae. So the cocci may be oval, elongated, or flattened on one side. The cocci remain attached after cell division. Even though after the cell division, some of these cocci remain attached. These group characteristics are often used to help to identify certain cocci. For example, in the case of diplococci, streptococci, tetrads, because they fail to remain separated after the division. That's why the shape like this are formed. First one, example for the diplococci are arranged, the cocci which are arranged in pairs. For example, Streptococcus pneumoniae, Moraxella catarrhalis, Neisseria gonorrhoeae. So these are the diplococci. I have shown the diplococci as well as streptococci. So both of them, the division is the plane of division. Look, the plane of division, both of them same. It has a one plane of division, and the symbol, not symbol, scanned electron microscopic view I had shown in the right side. So examples for diplococci are Streptococcus pneumoniae, Moraxella catarrhalis, and Neisseria gonorrhoeae. Next is about streptococci. The picture I have shown in the previous slide. The cocci which are arranged in chains as the cell divide in one plane. In the one plane, for example, Streptococcus pyogenes, Streptococcus agalactiaceae, etc. are the examples for streptococci. Next coming is tetrads, in which the cocci are arranged in packets of 4 cells. So the cocci are arranged in the packets of 4 cells as the cells divide in 2 planes. Example, Aerococcus, Pediococcus, and Teratogenococcus. So these are the examples of the cocci which are arranged in tetrads or in the packet of 4 cells, in which the cell divides in 2 planes. These are the examples for tetrads. Here we are showing 2 plane of division which give rise to tetrads. So that's all. We can wind up this session. So the rest part I will take in next class. Till then, take care and watch these videos. Thank you.